Hey everybody, I just wanted to take a little bit of time to go through some of the material that I missed out on last week with you. We left off talking about meiosis. So the big difference with mitosis and meiosis is that mitosis is all about cloning cells and cloning body cells, what we call somatic cells. But meiosis is a little bit different because meiosis is really just about sex. So it's about how organisms who sexually reproduce go about that in order to best fit their environment and best ensure species survival in the future. So one of the terms that we work on with meiosis is we associate the word gamete. So a gamete is just another word for a sex cell. For a mammal, it would be something like egg or sperm. If it were a plant, might be something like pollen and egg. So meiosis is really important in terms of sex because it does two really important things. It creates genetic diversity, so it allows us to have offspring that are all genetically different to each other. And then also it alters the chromosome number so that when an offspring is produced, it does have the correct number of chromosomes. So down here a little bit at the bottom, my fantastic drawing skills. The way we achieve this is we have one gamete producing cell. Let's say it's a human cell, so it would start with 46 chromosomes, which would be our diploid number. It would go through one division, where you would end up with two cells. But then what makes meiosis unique is that those two cells would divide again, and then we call that meiosis part 2, and then you would end up with 23 at the end. Now the way you end up with 23 is that there's no such thing as interphase, so there's no interphase part 2. So we don't want to copy the DNA again at this point because then that would bring us right back to 46. So instead we skip interphase 2. There's no such thing. And then we just move right on to dividing again. So this would be then our haploid number. We talked a little bit about fertilization, and obviously fertilization is kind of the source of all the variation we see in evolution. So this is a real picture through a microscope of many sperm seeking out an egg, and just kind of the moment that the sperm meet the egg. So ideally one sperm would penetrate the egg, and then what happens is there's a chemical change in the surface of the egg that hardens and therefore prevents any more sperm from entering the egg. I'm sure it's happened on occasion where two sperm would enter the egg, let's say, and now you have an odd, crazy number of chromosomes. So more than likely that would not even develop into any kind of embryo at that point. In the case of identical twins, in case you're wondering, identical twins is still one sperm and one egg. But then once the zygote situation happens and you've got your cell that's kind of dividing one cell into two cells into four, what will happen is that this will split off and you'll actually end up with two independently reproducing embryos at the same time. And they are genetically identical because their origins, the same sperm and the same egg, are what created them. In the case of fraternal twins or non-identicals, it's actually two sperm and two egg. So fun fact, many females release more than one egg every month. So sometimes it just so happens that both eggs could get fertilized. And then you'd end up with fraternal twins or fraternal triplets, something like that. Okay, so a little bit on why meiosis has to happen. So if we use mitosis to produce our gametes, our sex cells, what would happen in a human is you'd have 46 chromosomes in the egg meeting up with the sperm, who would also have 46, and then the developing zygote would have 92, and that's just not possible because it probably wouldn't even develop into any kind of embryo at all. Plus, the human chromosome number is 46. So what meiosis does is that it cuts this number in half. So it brings us down to 23 for both our egg and our sperm. That when they fertilize, then you end up with 46. So what we're really making here are haploid cells. So sperm and egg are haploid cells. So a little bit of details here about the stages. You can see meiosis part 1 and then meiosis part 2, where we divide once and then divide twice again. The colors here are representing maternal and paternal chromosomes. So let's say that red is the mom and the light blue color is the dad. So clearly this is not a full set. This is definitely not a human. There's only 4 chromosomes here. So there's not much going on, but for the sake of visual help, there it is. So what happens in meiosis is that you have 2 copies of every gene, and they're located on what we call homologous chromosomes. One chromosome comes from your mom, let's say, and one chromosome comes from your dad. And your parents might give you slightly different alleles, like mom gave me the dominant and dad gave me the recessive. So what happens in meiosis is that these homologous pairs will find each other and they line up and they cross over. There's a little picture of that on the next slide. So the color swapping you see is showing you like the random shuffling of genes from one chromosome to another. So you're just kind of swapping what's on the mom chromosome and what's on the dad, and then they divide and then they divide again, and you can see that at the end here we make 4 cells, and then each one has genetically a different combination of things, and that's what explains why you would be genetically unique to your brothers and sisters if you have some. So here's a little bit more about homologous pairs and crossing over. So once again, a very small number of chromosomes in this individual, but they would duplicate during the S part of interphase, and then we'd have this business going on. But they are containing the same genes, but just different alleles, different versions of each gene. So then when crossing over happens, they find each other and then they hug. They literally embrace so tightly that they swap pieces. Now, what pieces they swap is totally random and unpredictable. There are some things that we can predict, like the farther 2 genes are apart from each other on a chromosome, the more likely they are to cross over. If 2 genes are really close together, then maybe the chance that crossing over would happen here would be rare. So there's a way to calculate the likelihood of crossing over to take place, but that's a little beyond of what we want to do here. Also, this happens during the very first stages of meiosis part 1, during prophase. The synapsis part is the crossing over part that you see. A tetrad would be 4, like Tetris, a favorite game of mine, where you have 4 individual chromatids that are intertwining together. This right here is the reason why every egg that I make and every sperm a male makes is genetically unique, because every single time crossing over happens, it's random and it's new. So a little detailed here, they say that the chances that you could make any one particular egg with that combination, let's say, of genes is something like 8.3 million. So you are definitely more than 1 in a million. And then when you take into account you have any 2 parents in the world, and those 2, that egg and that sperm getting together, it's an incredibly rare event. Okay, so crossing over leads us then to 4 cells at the end, and they would all be haploid. Now for males, they will keep all 4, so they actually make 4 sperm. For females, it's a little bit different. We don't make 4 eggs at a time. We don't even have that many eggs. We only have a few hundred. So what happens is that some of the eggs become neutralized and then kind of feed into another egg. They become polar bodies. So that's why that egg in that previous picture was so huge. We just kind of suck up all the nutrients out of those. Okay, and then finally a little comparison between mitosis and meiosis. So in mitosis, we were taking 1 cell and dividing it into 2 cells, and all the cells we make are identical. The cells are diploid, so we have a little symbol for that, 2n, meaning 2 times the number of chromosomes, 1 from mom, 1 from dad. We do this when we want to grow in size, when we want to replace cells that are broken, and very importantly, there is no crossing over, and that's why all the cells end up being identical. Meiosis is all about sex, so we go through 2 cell divisions, where we start with 1 parent cell that is 2n, it divides once, and then it divides again, and they end up as 1n, or what they call just n at the end, and these are called gametes, and crossing over is what makes them unique. Okay, so I hope that helps, and I will hopefully see you soon. Bye.